Maybe I'll just get started on the introduction. So on behalf of the Wildlife Department, we'd like to welcome you to the this edition of the Next Generation Wildlife Ecoseries. Our Next Generation Wildlife Ecoseries highlights the achievements and amplifies the voices of early career wildlife biologists. This next generation of scientists is more diverse than the community of natural resource scientists as a whole currently is. We recognize that systemic racism continues to impose barriers for Black, Indigenous, people of color, BIPOC, people seeking careers in natural resources. And this speaker series will not only highlight professional achievements of BIPOC scientists who are role models for this next generation, but it will also serve as a forum to discuss how our field can move forward to reduce barriers and ultimately become a more inclusive community. The Wildlife Department's committed to challenging status quo in the discipline and working to propel a more diverse next generation of wildlife biologists forward. With that, I will turn it over to Frank, who will introduce today's speaker.
Thanks, Dan. So it's my pleasure to introduce Maybelline Gamboa, who's our speaker today, our last speaker of the semester for the Next Gen series. Maybelline is a PhD candidate who is broadly interested in understanding the evolutionary processes that facilitate and maintain adaptation in species along elevational gradients. Specifically, Gamboa aims to infer local adaptation in song sparrows that are distributed along a climate gradient on the California Channel Islands. There, she implements an integrative approach to understanding adaptation by combining genomics, functional morphology, and physiology. Ultimately, this research will assist in the identification of a source population for reintroduction to an island where this species, which is a species of special concern in California, has been extirpated. And then Maybelline is completing her doctorate from Colorado State later this summer and has accepted a position as a visiting faculty member at Colorado College in the fall, right? Great. So I'll turn it over to Maybelline.
Awesome. So thank you so much for that lovely introduction, both Daniel and Frank. I appreciate it. And I really appreciate your efforts and initiatives at Humboldt State University to really amplify the voices of historically marginalized communities within ecology. I identify as she, her with my pronouns. I am a proud Filipino American woman, born and raised in Southern California to wonderful, wonderful Filipino parents that migrated here in the 1980s. And I'm really excited to share my doctorate research, really just a small subset of my doctorate research. It's been a sort of a meandering path that focuses on how we can understand local adaptation and use that understanding to then inform animal reintroductions. And hopefully we have some time after the talk, so we'll spend most of my time presenting my research, but then afterwards I'd like for us to really discuss how we can use this information to actually apply it. And I think this is a really relevant department to ask this question, given that most of you are involved in some sort of wildlife management. So before I begin, it is Earth Day. Thank you for highlighting that. Here's a picture of our Earth. And one thing I do want to acknowledge at the beginning of my talk is that we're not scientists because we're knowledgeable. We're scientists because we're curious. And this is a quote from one of my friends at the department here. Pardon me. And I want this to be a seed that's implanted in you as you listen and engage with the material throughout the talk. I do have my chat function open. So if you have a talk or a question related to the talk or have a clarifying question at all at any point, go ahead and pop that into the chat and I'll go ahead and answer that time permitting. And again, I want us to be able to, I want this statement to empower you to engage in the conversation that we have afterwards, where we talk about the application of our knowledge of local adaptation for conservation efforts. So now switching gears completely to a more sobering reality of some of the factors that we're facing today in terms of biodiversity. So we know that we're experiencing immense global change that is leading to a lot of catastrophic events across the nation and across the globe. And in response to these really threatening forces, populations can either adapt, and that can be plastic or genetically based. They can disperse or go to these different environments, or they can go extinct. And many of us that study these very vulnerable populations are very aware of the fact that small populations are going to be much more vulnerable to extinction than, say, large populations. So I think many of us are also familiar with the extinction vortex process, but just to refamiliarize yourself, time is going to be moving in a downward fashion in this figure. And if we start with a small population, then that small population is going to experience the adverse effects of inbreeding, genetic drift, or the random fixation or extinction of alleles based off of the random sampling of individuals from one generation to the next. And that then leads to the loss of genetic variation, a reduction in fitness and adaptive capacity, and so on and so forth until we can spiral down into a smaller and smaller population. And historically, when conservation management teams have really looked at trying to manage these small populations, we've often focused on establishing corridors and looking at movement, looking at behavior, and looking at demography. And when we talk about genetics, most of our genetic understanding has focused on neutral genetics. So looking at the effects of inbreeding, looking at genetic drift, looking at the loss of genetic variability at these neutral genomic markers. And that's because largely we didn't have the tools available to us to actually answer these questions related to adaptive potential at the genomic level. And so with the advent of next generation sequencing in the past 20 years, we've really been able to focus more so on this and focus on it in a way that allows us to inform our management more effectively, especially in the context of reintroductions and translocations. And so let's just go ahead and set the stage for why this might be important in the context of these conservation efforts. And so adaptive traits, again, are going to be any sort of trait that increases fitness in a particular environment and are favored by selection. So let's just take this toy example of these 3 different habitat types. Overlaid onto those habitat types are different color matched birds. And that color, let's just assume, is adaptive, such that individuals in a particular habitat, if they're color matched, will have a higher relative fitness compared to individuals that are not color matched. And let's say we have another target environment in which we want to introduce or translocate individuals to. And if we were to introduce all of these different colors into that environment, according to some hypotheses, like the pre-existing adaptation hypothesis, the ones that are going to succeed and survive to the next generation, if color is adaptive, are going to be the blue color matched birds. And so understanding adaptation is really important. And it's also really important to understand if that adaptation is actually genetically based. We know that other processes can result in adaptive variation, but when those adaptations are genetically based and incur some sort of fitness advantage, then individuals that have that particular adaptation can be favored by selection. And what we see are evolutionary changes. So again, thinking about this from a genetic perspective, if there is one gene that controls color in the system, and that color is biallelic at that gene, such that big A individuals have that blue phenotype, then we would want to target genotypes that have big A in order to reintroduce them to that blue target environment. But as with all ecology, not all traits follow the same sort of simple explanation. So some traits are not examples of local adaptation. As I mentioned before, you can get adaptive variation through a number of different processes. Also, not all traits are products of natural selection. They could be products of other neutral evolutionary processes. So for example, one of the most common mechanisms in which we can get adaptive variation is through plasticity, in which the environment acts to create different phenotypes among individuals that have the same genotype. Alternatively, as I mentioned, other neutral processes, like say genetic drift, can result in the variation that we see today. So for example, we know that the shape of our noses don't infer some sort of fitness advantage. That is to say, the shape of my nose doesn't necessarily dictate or isn't significantly correlated with how many offspring I have that survive to reproduce to the next generation. And so the shape of the nose itself is a neutral trait. It has nothing to do with my fitness in general. But if you were to be some sort of extraterrestrial organism, for example, and you subsample individuals from the global population, and all of those individuals happen to have noses and happen to have eyeglasses, then you can come up with this false assumption that the shape of the nose is intended to uphold your glasses. We know that's not the case, clearly, but in decades of adaptation research, many ecologists fell into this trap of assuming that phenotypic correlations are evidence of adaptation, and they're not. And so Gould and Lewontin formalize this more specifically in this very seminal paper in evolutionary biology, in which they say they use the analogy of spandrels. So spandrels are going to be these intermittent sections here that are created through, as byproducts rather, of these adjacent arches next to each other. So it's the arches that are actually providing a lot of the structural support, and these spandrels just serve as this sort of empty space. And in order to fill that empty space in this cathedral, there are these superimposed images of, say, saints or landscapes or what have you. But the fact that there is an image there does not affect the structural integrity of the actual cathedral. And so again, we have to try to avoid these spandrels in adaptation studies, or this false misconception that some sort of correlation between phenotypes is evidence of adaptation. And so broadly, as Frank mentioned in his introduction, my research is really motivated by this question of what generates and maintains phenotypic variation in natural populations. And I answer this from an evolutionary perspective. So what are the evolutionary mechanisms that interact to generate this variation? And is that variation actually adaptive or non-adaptive or neutral, or is it evidence of local adaptation? And I couch this under this entire umbrella of conservation, because I think that these types of questions are really pertinent for answering other similar questions related to conservation management of these populations, specifically in the context of adaptation. And I mentioned that genomics are really amazing and that they allow us to identify these punitively adaptive loci now across the genome. But they're also a double-edged sword. So much like we have these spandrels, this false misconception of adaptation based off of the correlation of phenotypes, we also have what's called molecular spandrels. So in other words, the misconstruing of adaptation using molecular evidence based off the correlation between genomic data and phenotypic data. So sort of this genome-wide association framework. Genome-wide association tests are useful, they're just not strong evidence by themselves. And so in order to really establish this, I use this integrative framework in which we link the environment or the hypothesized agents of selection with the phenotypes or the hypothesized agents of adaptation with the genotype. And by establishing these links, we can create a more holistic understanding that allows us to infer local adaptation in natural populations. So for example, we can find evidence for selection acting on individuals that exhibit trait variation. We can take a population genomics approach, analyzing that genomic data to estimate the relative strength of drift and potentially driving variation. We can also use that genomic information and relate it to our hypothesized agents of selection to infer the link between the environment and genotype. And then again, applying genome-wide association tests to really find evidence against plasticity. So is there any genetic basis to this trait? And I've done this over the years in a number of different systems, or helped out in a number of different systems. I've helped out with the island scrub jay research, looking at microgeographic adaptation. We've looked at different data associated with swamp guppies in the Gamboa lab. And more recently, I'm also co-lead on another project where we look at adaptation in response to different elevations and different shrimp at different lakes. But most of my research revolves around this very beautiful, charismatic, wonderful brown bird called the song sparrow. And I know everybody is so elated to discuss this magnificent bird, but you'll have to contain yourself because we do have several pictures of the bird throughout the talk. And even though they are widespread across North America and exhibit a lot of phenotypic variation across that range, we're going to be specifically focusing on Channel Island song sparrows. And in that, we'll look at song sparrow bill divergence. I've looked at a number of different traits as well. So I've looked at traits associated with plumage and physiology, but for the interest of time, we're just going to focus on the bill for a bit, and I'll touch on those other traits and the findings of those studies at the end.
The sound anyway.
So now moving forward, our question then, is there evidence of local adaptation in Channel Island song sparrow bill sizes? So just to preface this, song sparrows historically were on eight Channel Islands. And on 2 of those islands, they were extirpated, and those extirpations are directly related to human effects. So on Santa Barbara Island in particular, which is owned and managed now by the National Park Service, the restoration efforts are at a stage in which we can really start to think more realistically about the reintroduction of song sparrows to that region. And so in collaboration with Nature Conservancy, Smithsonian, Park Service, and CSU, what we're doing is we're trying to develop ways in which we can use our genomic understanding to inform the reintroduction of birds to this island, where they were lost because of introduced rabbits. And I know they don't look that terrifying, but they completely destroy habitats for a lot of songbirds. What's also interesting about this particular population and this distinct subspecies is that they occupy a really strong climate gradient. So on Santa Cruz Island, it's very hot, arid, still, it's very xeric, very analogous to the interior of Southern California. And then as you move west, what you see are conditions that are very cold, wet, windy, very overcast. So more analogous to say, the Redwoods region or the Pacific Northwest with winds of up to 60 miles per hour on any given day. And so across this really small spatial extent of about 100 kilometers, we find a climate gradient that really spans the latitudinal extent of California at least. Another thing that's really important, especially when we're talking about hypothesis testing, is the bill as a multifunctional tool. So much like our hands, the bill is involved in preening and communication and mate acquisition, and probably most notably associated with foraging. And song sparrows themselves are pretty generalist species. They forage on insects and fruit in the breeding season and switch to seeds and grains in the winter seasons. And the reason why most emphasis has been placed on the bill as a foraging tool is because it's been studied so extensively in the context of Darwin's ground finches. So today we will focus a little bit on the foraging ability, but we'll also focus on this alternative hypothesis, and that is that selection is operating on the bill to facilitate thermoregulation. So much like an elephant or a jackrabbit's ears, birds can shunt blood to this vascularized region of the bill and dissipate dry heat. So much like so rather than relying on evaporative water loss, which results in a loss of heat, but also a loss of water, birds can actually, in these arid regions, just shunt blood to this radiator, radiate out heat, maintain appropriate water balance in the interim as well. So again, we're going to look specifically at the foraging hypothesis and the hypothesis that says selection is operating on the bills to improve thermoregulatory ability. And again, we're going to be testing this a little bit indirectly just because we didn't have the resources to look at this more explicitly. So to do this, we measured bill surface area across the 3 main islands And this could not have been done without the help of many wonderful field techs, one of which is a student here in your cohort, Janelle. She actually provided the brunt of a lot of the sampling for this effort. So thank you for those 300 sparrows you sampled with me. And a number of other candidates, field techs as well over the years. And they've helped me not only with the sampling effort, but also with my physiology experiments as well. And so in this particular system, we measured bill surface area as it relates to the length, the depth, and the width of the bill. And what we find is we confirm patterns that have been found in historic museum specimens. And we confirm these patterns in these contemporary populations where raw bill surface area here, and also bill surface area corrected for body size, which we'll talk about later, are actually significantly different between these islands. So then this begs the question of why? Why might they have different bills? So when we look at all these different territories and these unique sampling locations, and we really try to parse out what birds might be eating in these different regions, we decided to use vegetation type as a proxy for available food resources. This study wasn't necessarily designed to adequately test this, but this provides us with a really good stepping point to move forward to then look more closely at vegetation and food resource differences. So broadly across the different islands, they occupy these grasslands interspersed with a lot of low shrubs. And in our analysis, what we did is we categorized the abundance of these different focal vegetation types. So different grasses, sage, fennel, coyote brush, lupin. Those are all dominant shrubs in these regions. And we also had miscellaneous herbaceous flowering plants, and then other was water or bare rock or bare ground. And so based off of these ranked abundances, we were able to create a contingency table relating these ranked abundances of vegetation types and the different islands, and did find a significant association between those two variables. And that's not surprising because we know climate affects vegetation to some capacity. But when we plot this then and reduce this complexity of these highly correlated traits into multivariate space using nonlinear principal component analysis, what we end up with are two axes of variation that explain about 50% or so of the variation observed in the vegetation. And on the primary axis of variation, we find grasses explaining one side of it and sage, lupin, forbs, and other combined explaining the other, such that there's this trade-off between the two. And then on the second axis of variation, we see this trade-off between fennel and coyote brush. So those are our most dominant shrubs in these territories. And so plotting then our individual unique sampling locations in this multivariate space, what we see is an extensive amount of overlap. And again, that's not surprising, given how broad our categories were, but we also see clear separation between Santa Cruz and some of the other islands, primarily on the second axis of variation, in which Santa Cruz Island territories were typically had these introduced fennel in their territories that they were using. So again, to look at this a little bit more, we would need to design more specific sampling of these resources, especially in the non-breeding season when they actually use those food resources. But it's a great starting point for us. And when we condense that vegetation space into different coordinates and then relate those particular coordinates in PC space to the individual phenotypes, and then also combine that with the maximum temperature in those sampling regions, we can run a multiple regression analysis in which we look at the effect of vegetation dimension 1, vegetation dimension 2, and climate, or maximum temperature specifically, on predicting bill surface area. So here we're using residual bill surface area, or bill surface area corrected for structural body size. And then here I'm plotting only maximum temperature. And that's because when we run these multiple regression analyses, we find that actually the vegetation dimensions explain almost none of the variation in residual bill surface area. Yet we see this very significant effect of maximum temperature. So there's some evidence that suggests that maximum temperature is driving this, but we interrogate it a little bit further and look at these foraging efficiency traits. So taking more of an experimental approach in the field, what we did is we sampled individuals and brought them into these enclosures, and we recorded them foraging on a standardized food substrate. We also took individuals in and looked at the bite force as it relates to the bill size and bill dimensions and to body size. And here's sort of the 2-minute takeaway from that. There's no difference in bite force or seed extraction time between our two extreme islands. So here we sampled primarily San Miguel, shown in blue, and Santa Cruz, shown in red. And what we find with maximum bite force is that even though there are these distinct bill differences and separation on bill depth space on the x-axis, we don't see any effect on maximum bite force. And when we look at other bill dimensions and body size as well, we still don't see an effect on maximum bite force. Similarly, if we look at more than just length, depth, width, and all these other characters, if we look at a more complex measure of bill variation in PC space, what we see is, again, separation between Santa Cruz Island, where Santa Cruz birds tend to have these like broader, more wider bills, and then San Miguel birds tend to have these more narrower, sort of pointed, slightly longer bills. We see that there's that difference, but we don't see any differences in terms of seed extraction time when controlling for individual variation using linear mixed models. So clearly there is some sort of effect of climate. Our vegetation dimensions weren't able to find some sort of direct relationship necessarily between vegetation dimensions and bill surface area. And we don't see any effect of those bill differences on key foraging efficiency traits. So that suggests that then we find evidence for bill size that's related to climate. So now we wanted to look more so at the genomics underlying that. So here, let's just, here's a sampling map of our genomics. And across the 3 main islands, we took blood samples from individuals, and we also took blood samples from individuals on Anacapa Island, where they recently colonized, and from the mainland. So the mainland serves as sort of our outgroup, so we can really parse out the fine scale variation among our islands. And here I'm going to present proportion ancestry in sort of a structure-like plot. So proportion ancestry is going to be on the y-axis. Each individual is going to be presented as a bar, and the relative amount of their genetic variation that is attributed to a particular cluster based off of this algorithm is going to be shown with different colors. So underneath all the individuals, we have San Miguel birds grouped here, Santa Rosa, etc. And we see that there are clear, distinct patterns in these neutral markers. So using a reduced representation approach, we generated 22,000 neutral SNPs to create this genetic clustering, this estimate of population structure, rather. And we can confirm that, again, with other algorithms. We find congruence among these 2 different algorithms that use different methods. And that's on the primary axis of variation, the mainland, again, is distinct, and the islands separate as well, with San Miguel being the most distinct. And it's also important to note that we did recover some migrants in our sampling, which was surprising to us, and I'd be excited to talk to somebody about that afterwards, because this is really interesting evidence of potentially exploratory behavior. And also, I'd be happy to discuss with anybody that's interested in this, the fact that there is more admixture on Santa Cruz Island. So based off of this, we know that there are neutral genetic differences. So our bill difference is just a product of genetic drift then. So here I'm going to present genetic variation as effective population size. And based off of evolutionary theory, we know that if effective population size or genetic variation is large, genetic drift then is going to be relatively weaker as an evolutionary process in facilitating change. Whereas if effective population sizes are small, then genetic drift is going to be more effective at resulting in evolution, and then selection would be less effective at resulting in evolution. So let's go ahead and compare our effective population sizes here. And we see that they're actually relatively high, surprisingly high for an island subspecies, especially when we compare them to the Channel Islands fox, although the island fox has undergone a bottleneck. So we've also compared them to island fence lizards that have also been distributed across the archipelago. And again, among all these island species that are distributed widely across the archipelago, song sparrows have the highest genetic variation. So is it a product of genetic drift? Not likely, but again, we need more evidence that it's selection driving this. So using a number of different algorithms, we identified punitively adaptive loci. And so I'm going to present that data as just a series of Manhattan plots where chromosomal position is on the x-axis. And then Ken asked, do you mean inter-island or continental migrants? So with respect to those other migrants, I was specifically talking about inter-island migrants, yes. We didn't find any evidence of gene flow between the mainland and the islands. So here, chromosomal position again is on the x-axis. These are Manhattan plots, so we're going to have some sort of significance threshold on the y-axis. Because we're using a number of different algorithms to demonstrate this, anything that is above a red dotted line is evidence of a locus that is significantly associated either with that environment or significantly different among the populations based off of the population genetic structure that we recovered in our genetic clustering algorithms. So using both differentiation-based tests, so that looks again at the population structure, controlling for that, and looks at the outliers, and using genotype environmental association analyses, so relating maximum temperature to genotypes, we find a number of different loci, again, that are highly elevated or significantly associated with these factors. And so when we look more closely at these different loci, we find that in their annotations, they are found in genes or closely linked to genes responsible for craniofacial development. And those are pathways that have been well described to change bill structure in Darwin's finches, as well as in chickens and ducks. And they also control craniofacial development in mammals. So then returning back to this framework, when we look at the link between genotype and environment, when we only look at population genomics, we find that it's not likely due to genetic drift based off of our large effective population sizes. Instead, we find evidence that it might be linked to selection, specifically selection from this maximum temperature climate gradient. So then to really complete the picture, we need to see if there's congruence among these different algorithms. In other words, are the loci or genes or pathways that we recover in this first analysis relating genotype and environment the same ones or similar ones that control bill development when we specifically link the phenotype or the bill to the genotype? So here again is where we apply our genome-wide association tests. Everything is shown as a Manhattan plot, just as in the previous one. We use multivariate and univariate approaches, and using combined approaches or a hybridization of these two approaches to estimate heritability based off of our SNP data set, we found that 34% of the variation in the bill is controlled by the variation at these particular loci. And then again, when we look at the function of those loci, we do find evidence of selection operating on those or of the same pathways controlling bill variation. So as I mentioned before, what we're recovering is this congruence among a number of different ways in which we can infer local adaptation. And it's through the application of these different methods that we can develop a strong body of evidence to say, these populations are exhibiting adaptation. How can we move forward with this? And so really, this framework that I've used today helps us take the risk out of using molecular data in inferring adaptation by providing, again, that strong body of evidence. And that makes this much more approachable and applicable for non-model organisms, rare organisms, organisms with very few genomic resources, and organisms with complex traits. And I mentioned that I looked at this in a number of different traits, not just in bill morphology. Thermal adaptation itself is an emergent property. We often try to use some sort of one trait as a proxy of population responses to various environmental gradients. But instead, we need to take a more holistic approach and look at multiple traits to see how whole organisms actually respond to these environmental changes. And so again, we looked at this in feather structure, feather microstructure. I've looked at this in plumage coloration. And I've also looked at this in terms of physiological responses to temperature stress. I won't have time to talk about a lot of this today, but we also used our genomic resources to develop molecular baits so that we could target in museum specimens these same regions that have been shown to be elevated among these populations to see if those differences in bill morphology in museum specimens also track differences at these genes over time with climate. So briefly, I'll just say that in looking at feather microstructure and plotting those islands in multivariate space again, what we can see is Santa Cruz Island birds tend to have these airier sort of plumage barriers. So instead of having these really developed pennaceous sections right here, those are the ones that overlay on top of each other and are more exposed to the elements. The downy sections or the plumulaceous sections are going to be the ones that are close to the body, so those aren't directly exposed to the elements. But in Santa Cruz birds, this region, the pennaceous section, is less well developed. And it seems to be that more energy has been allocated to length instead of density of the barbs there. On the mainland, we could say, they wear something like a nice, strong, sturdy jacket. And then on Santa Cruz and San Miguel Island, the most interesting one, I think, pattern we saw in terms of these different feather structures is that they have much denser barbs, so a tighter weave, you could say, and more emphasis on developing the pennaceous section as opposed to the plumulaceous section. So what that suggests is that San Miguel and Santa Rosa birds, those birds that are experiencing those really windy conditions, those really wet conditions where you get a lot of dew that can build up on your plumage, we have birds that have feathers that are overlain tightly on top of each other, such that it creates a really strong barrier against wind and water. And then when we looked at these physiological traits, we looked specifically at metabolic rate, which I won't talk about today, but we also looked at the critical thermal limits. So endotherms, much like us, or endotherms, including us rather, follow this sort of Scholander curve in our metabolic rate in response to temperature, where there are certain limits of temperature that we can tolerate without having to exert any energy to maintain homeostasis. But past those limits, so if it's too cold or too hot, then we start to really use our metabolic rate to modify our internal temperature. And so I'm going to switch the axes here so that temperature is on the y-axis now. And what we see is, sorry, I don't include the sample sizes on here, but among a number of different experiments, we find that on Santa Cruz Island, they have elevated upper critical temperatures, and that's significantly different from the upper critical temperatures on San Miguel Island. And this in itself is a really remarkable pattern because we don't often see these differences at population levels, and we don't see them at such a small spatial extent. So what all of this together means is that populations are really responding to this strong climate gradient by adapting in a number of different ways. And in order for us to move forward in terms of management, and especially in the context of biophysical modeling or niche modeling or climate modeling, we need to really consider all these traits when we're thinking about the adaptive potential of populations. And then I'm going to go through my acknowledgements and funding slide and then open it up for discussion if we have time, more so about some of the ways that we could take this information and move forward with it. First and foremost, and this should be absolutely everybody's first thought when they're thinking about doing research on any lands in North America or across the globe really, is that the Channel Islands and much of California were stewarded originally by the people of the Chumash Nation, and I acknowledge and honor and respect them, because without their contributions to the stewardship of that land, we wouldn't have the biodiversity that we see today. And so I just wanted to highlight that and also highlight my committee members and our funding sources. Again, this is a collaborative project among a number of different agencies. I'm happy to take any questions about the research before we go into the discussion. But again, you can also find me on Twitter, and then my website is there as well. And so I'd be happy to take questions now, or I can go ahead and move forward with the discussion. So if you do have questions, you can go ahead and paste that in the chat. And so while I'm walking through some of these really sticky points that I've been really thinking about towards the end of my dissertation, then if you do have that question and it pops up, I can go ahead and address that as well. So most reintroduction translocations have occurred in really charismatic megafauna. So think mountain lions, think wolves. These are animals that are really heavily funded, that have a lot of public support because they're so charismatic. Most reintroductions have occurred in the animal systems, have occurred in avian systems. Here's the Lincoln Park Zoo's database on documented reintroduction efforts with either wild populations brought into captivity or captive populations. And what we see is that among the thousands of vulnerable species and populations across the globe, we really have only managed to do this in about 200 species that are documented. And so reintroduction and translocation efforts are themselves a very niche concept within conservation efforts. And when we're talking about reintroduction efforts, I don't want us to divert away from neutral genetic diversity. I think neutral genetic diversity is still very important in conservation genetics. And it's really surprising that we're still getting papers like this published a couple months ago in PNAS that say that we don't need to study this. Here in this entire talk, I focused on adaptive genetic diversity, but we're really doing our organisms a disservice by only focusing on that. So I think that we should be critical in looking at this type of data and acknowledge that these two need to be complemented. And so then that brings me to the question then of, now that I have all this data for these projects, how do we move forward? And it also begs the question of, in the context of all the potential conservation efforts, we're faced with managers that are trying to ask us or trying to get us to justify why this is important. And increasingly, I keep telling myself that conservation genetics is so very critical to all management strategies. But then we still get these papers that come out that say that the field of conservation genetics is rooted in these misconceptions. And so I'd be interested to hear how people in the wildlife department here have approached these conversations with the managers that they collaborate with. And again, I'd be happy to take any questions as it relates to my research, as it relates to a lot of the efforts that I've done actually at CSU in developing workshops, spearheading different initiatives, and developing training sessions for graduate students to improve diversity, equity, and inclusion within the ecology program and within higher education in the biology department. So thank you so much.
Thank you. That was awesome. Really very well done. I'm very inexperienced in all of this stuff, but I think this was great. Thank you.
Thank you. I appreciate that.
Yeah, so if you have questions for Maybelline, you can either raise your hand or type them right into the chat, or if you want to add to her her discussion points, please feel free to do that in addition to questions.
Yeah, Barbara.
Hi, yeah, great talk. I was wondering, the differences you saw across the islands. I think there was on a slide I saw ancient DNA. I think I saw it somewhere.
Yes.
Because I was thinking, like, could there have been some founder effects, like of the individuals that ended up on those islands that came with particular traits that could have contributed to some of those differences?
Potentially. We used our mainland population as sort of an outgroup. And so when we were developing our panel of potentially adaptive loci, we did include different analyses, including the mainland and just the island specifically. When we include the mainland, we don't find any patterns like that associated with it. Again, those genetic signatures that could have been founder effects from the mainland population might have been washed out in contemporary mainland populations. So that is a factor to keep in mind as well. We included in our panel some museum specimens of mainland populations as well, though. So you'll have to wait and see what that data looks like, but it is an exciting follow-up to what we've found so far.
Yeah, great, thanks.
Yeah. I have a question that says, how do you think migrants are affecting the genetic variability of island populations? So typically we're getting, so I should also say song sparrows are really poor flyers. The evidence that we see of them being able to fly actually across these water barriers is pretty surprising, especially because there is this really strong wind gradient. So it makes sense that we have admixture on Santa Cruz Island because the wind funnels in such a way to push birds sort of in that direction. What's more surprising is that we get those migrants from Santa Cruz Island sort of exploring seemingly Santa Rosa and San Miguel Island. And we haven't followed that up with any analysis of parentage or further genetic analysis of this next generation of song sparrow populations, but most likely that's just exploratory behavior. In terms of gene flow between the islands, if there are individuals that move from San Miguel, Santa Rosa to, say, Santa Cruz, then that can sort of wash out some of the unique signals that we see on Santa Cruz Island. So I'm not sure if that quite answered your question, but genetic migration and gene flow in general tend to sort of normalize or homogenize that genetic variation between the islands. Yeah, Daniel.
Well, I think Frank had his hand up first.
Oh, okay, Frank. Yeah, sorry.
I'm going to sidestep your genomics question for a moment, just because it's a tough one. You mentioned the latitudinal gradient, or the temperature gradient on the Channel Islands is kind of analogous to the latitudinal gradient in California. Have you all looked at whether any of these phenotypic characters you're looking at vary in a similar way on the mainland, song sparrows being so widespread just throughout the North America?
Yeah, absolutely. This has been looked at in museum specimens, and it's pretty well described that there's that clear pattern of bill variation with respect to maximum temperature in California and on the islands. And that's something that my collaborators, Ray Danner and the late Russ Greenberg formalized in a 2012 paper. But looking more broadly out at patterns across the entire song sparrow range, it's really difficult to answer that question in contemporary populations because we don't really have any individual research programs that are looking specifically at those phenotypes across their entire range. But again, we do find evidence of similar patterns in museum specimens as it relates to plumage color, bill differences, and no one's looked at feather microstructure in these museum specimens, but that's definitely something that I'm hoping to get an undergraduate at Colorado College to think about, hopefully.
Thanks.
Mm-hmm. Yeah.
Just a great talk, just really awesome work. And I mean, you've made the most of these song sparrows in so many ways, but it raises so many interesting questions too,
Yes.
right? So one that I was thinking about was you've got this strong east-west line in climate that's broken up into 3 distinct units, demes, if you want, of
Yes.
individuals inhabiting these different islands. And like within an island, you have an interplay between perhaps habitat selection, where, you know, birds that are wearing a light jacket but maybe are better adapted to a hot environment might gravitate towards the east side of the island, and, you know, ones that are wearing a fur coat might gravitate towards the west side of the island. So there's this sort of interplay between local adaptation, natural selection, and habitat selection of individuals based upon their physiological preferences. And I mean, I may have missed it, but how does that really show up in your data? Because the between island differences were very clear. But how about the sort of within island differences?
Yes, so we did look specifically at within island differences as well. And so my dog is sleeping and seemingly attacking somebody in her dreams. But when we look at these within island differences on Santa Cruz Island, there seems to be so much gene flow across that island that a lot of those signatures are swamped out. So again, it's very clear when we compare Santa Cruz broadly as a group to other islands, but when you compare individuals from, say, the western end of the island to the eastern end of the island, the patterns tend to get washed out, and we actually just get a lot of variation across that gradient, across the intra-island gradient, rather.
So is that, do you think, I guess what I'm really getting at is, do you think that that's caused by a failure of those individuals that are locally adapted, in a sense, to a particular thermal environment to actually sort of track those habitat features? Because, I don't know, you know, there are other reasons they might disperse, like inbreeding avoidance or available territories, and that sort of counteracts that thermal adaptation.
Yeah, absolutely. And that's definitely a question that came up in terms of something I wanted to explore further, but again, in the context of your dissertation, how many questions can you really address? So it's something that I'm interested in, because what we're seeing is that on Santa Cruz Island, over the years, they've actually increased pretty dramatically in population size. And they only recently colonized Anacapa Island in the 2010s or so and established a consistent breeding habitat. So even then, people thought that maybe Anacapa and Santa Cruz were actually a subset of the mainland, but our genomic evidence suggests that they're not, clearly. And what I think might be happening is that on San Miguel Island, they have really great territories there, really great habitat for them. On Santa Rosa Island, again, they have really great habitat for them. But it almost seems like this movement sort of east is because they're at carrying capacity in those systems. And on Santa Cruz Island, we've seen, again, an increase in those population sizes, but we're also getting really, really strong competition between song sparrows and Buick's wrens. So if anybody wants to do a project looking at the competitive interactions between song sparrows and Buick's wrens that use the same habitat space, then I'd be interested in talking more about that later. But yes, there's just so much more complexity within Santa Cruz Island that you could really generate an entire dissertation project just looking only on Santa Cruz Island. Yeah.
I think Janelle had her hand up.
Yeah, Janelle.
My turn.
I will reiterate, this was a great presentation, May. It's
Thank you.
also really cool to go from working on your project in 2015, pulling feathers and collecting blood, and seeing the results of that.
Yeah.
So I remember, and I've talked about this in ornithology a bit, so a lot of the undergrad students here have probably heard me mention this a couple times, but when we were using playback to capture the birds, the Santa Cruz birds, we used the organ song. And then I remember using the New York song for the colder, windier San Miguel. And I'm wondering if you or anyone is doing anything with that.
So that is a great question. I don't know if my screen is still shared right now. I think it is. So I actually had one of my former field techs investigate that question, do a preliminary analysis on, she couldn't get to San Miguel Island, but she took some preliminary recordings of Santa Cruz and Santa Rosa birds. We took measurements of their bills as well. And what we found are in line with the literature that relates bill sizes to song changes. We find differences in trill rates, but again, this was just a subset of individuals. And so she talked with Mike Webster at the Cornell Lab of Ornithology about this project, and she received honorable mention for her NSF GRFP proposal, ended up taking another dissertation project in looking at aquatic insects instead, so we lost that one. But what was great is that Mike then recruited another student, Zena, who is still working on the islands, and I've helped mentor her sort of transitioning some of what I've learned to her project to help her develop her project a little bit more, but she's looking at this question specifically. So I couldn't answer it in the context of my time on the islands. And there are some other questions that I still want to ask as well, but Zena hopefully will be able to tell us more so whether or not those different songs or responses to different songs are related to these different bill differences as well. Yeah.
Cool. I feel like I just set you up for your next slide.
Thank you.
Do you know if she's going to do any translocation studies, like taking San Miguel birds to Santa Cruz?
So because this is the species of special concern, the Park Service is very wary about us, in particular, moving individuals across the islands unless we have some sort of justification for it. There are other projects that we've proposed with them. I've proposed looking at differences in muscle mass, looking at hemoglobin levels, looking at internal parasites, things like that. But it's really just how many times can I probe them until they're like, hey, actually, no, these are organisms that are protected. So yeah.
So I'll ask one more just to fill space, but to change subjects to something that you asked about that I think is super interesting. So, thinking about how we use the type of information that you've created here, right, and shared with us in planning reintroductions.
Yes.
A lot of the work, at least that I've been tangentially involved in, and one that a project that one of my grad students is working on right now, often the question is, how do we effectively supplement existing populations? And of course, there's the risk of diluting local adaptation by adding novel genotypes sourced from elsewhere in those reintroduction populations. So I guess, a general question or just discussion point is, what have we learned about that problem? And knowing that you can't experiment with it in this situation necessarily, right? But what have we sort of learned about that problem, or what would you suggest to someone planning a supplementation project based upon your work with song sparrows?
Yeah, absolutely. And it is really difficult because we don't have a lot of systems that have actually documented evidence of outbreeding depression. Fracom has done some reviews trying to establish some sort of data set for outbreeding depression, but really we don't know a lot. And when we actually run simulation models, or at least the simulation studies in the literature suggest that outbreeding depression is going to be something that is most likely very rare, because a lot of these traits are controlled by many genes of relatively small effect. So you'd have to have the right combination, right bad combination almost, in order to really have some sort of significant effect on fitness. And then actually your best bet, and what I think is conservation genomics is doing more so with neutral genetics, is identifying the most dissimilar populations in a lot of ways so that you can supplement them with individuals that have genetic variation that's different. Because ultimately, we're trying to superimpose our predictions about how selection operates when selection's not forward thinking. And so what might be adaptive in my study might not necessarily be adaptive given future climate changes. So really, we should be trying to create populations that are robust in their genetic variation, just so that they're more able to adapt to novel environmental situations. But again, that's my personal opinion. So and that is also very strongly related to my advising group as well. So.
Well, it gets into a philosophical point too, which is,
Yes.
should we be trying to conserve local adaptation or, rare genotypes in small populations, independent of the existence of outbreeding depression, right? So perhaps because that's just a novel phenotype, and the trout people have really contributed a ton here. But the world could be all rainbow trout, and we would still have trout, but we would lose all of that local adaptation and variation, right?
Yeah, absolutely. You bring up a really important point that is very heavily debated in the conservation biology literature, and in general in ecology. Where do we draw the line between what's a distinct population versus what's not, and what warrants actual protection? And really, I think, as scientists, we can inform the public and inform managers and stakeholders of the body of evidence that we find. But ultimately, it's going to be a combination of public opinion and stakeholder interest and a number of other factors, cultural importance and significance, that might not even directly relate to what our recommendations are. So I don't have a clear line. I think that that's very context dependent. But again, it's hard, and it's going to depend on that system and who's involved, how charismatic they are. Like we're talking about wolf reintroductions in Colorado, and it's very intense. Yeah.
Other questions for our guests? Sorry, I asked too many.
Thank you so much for your questions, by the way.
I guess, with that, we can thank you, soon to be Dr. Gamboa, for her time and her excellent talk again. And I'll point out, this does conclude our planned next generation Ecoseries for the semester. There are some other great talks going on in some of the other seminar series around campus, so look for communications about those and invitations to those. But once again, thanks again for your time, and it was great having you.
Thank you so much. I really appreciate it. Yeah.